So this is my title, What's Next After Next Generation Biodiversity? And it's really, what it really is ecology. So we talk here a lot about systematics and we want the genomes to improve our trees, but I think the people who make more use of the genomes and actually the ITS sequences and everything else are actually ecologists. And so the first thing I want to show is I think, an amazing slide that I don't believe is published yet, and it's about sampling. Ecologists are all about sampling. And this is from Noah Fear, who's at the University of Colorado. And this is a map of the distribution of an undescribed alternaria over North America. And what Noah did and his colleagues was to send to 1,000 citizen scientists in the US a kit with 2 swabs, and asked them to swab the outside of the door frame of their front door and with one of them, and the inside of the top of the door frame with the other one. And then they sent him back to him and he sequenced them. So he's got 1,000 of these. Now, the resolution with only 1,000 samples isn't very good, but you can see we could actually have range maps of our fungi, just like ornithologists have or botanists have, something to aspire to. But I'm going to talk more about not just where the fungi are, but what they're doing. So after community composition and range maps, what? And the thing is function. That's what ecologists now are really interested in. And you know, ecology isn't just the forest, it's guts, it's skin, it's everything. And so function, people in my department at Berkeley, most of them are interested in function, but they're not interested in ecology. They're interested in the function of one little mechanism. But the function I think that we serve as systematists is the function of fungi in natural communities. And so I first want to talk about the challenges, sort of the depressing part, and then we'll get to the successes, okay? And I'm going to mention 3 recent review articles that I relied on to make this talk. And the first one was by Kabir Pei on natural history and the way forward in modern fungal ecology. And he basically goes through why it's going to be hard. So first he points out, we know the identity of most operational taxon units used in molecular studies only imprecisely, and the usual blanket 97% sequence similarity cutoff used to delineate OTUs may obscure meaningful ecological differences. So from my own interest in Neurospora, I know that there are about 40 species of Neurospora plus these 5 or 6 of Sodaria at the bottom, and they've all got, their ITS has got 98% similarity. So when you get an ITS sequence from one of these ecological studies, it could be any one of those. So Kabir's right, it's imprecise, we need something better. And then he points out that the relationship between gene abundance and organismal abundance is not direct. So if you do an ecological study, you get back these reads, ITS reads, and you'd like to be able to say, well, the fungus that has a lot of ITS reads is the common one in my sample. But turns out that's not the case. And Anthony Ammon, some years ago in Tom Bruns's lab, did a very good experiment. He took an environmental sample and split it into 5 aliquots. So he had the same amount in each of those 5. And then to that sample, he added these 6 fungi here, spores of these 6 fungi. And he added them he didn't add them just in one concentration. He did a thorough job, so he added them in 1,000,000, 100,000, 10,000, 1,000, and then 10. And so, and they were spores, and then he treated the samples with these added fungi that shouldn't have been in the indoor air fungi. He was studying indoor air, but the fungi that he added were mycorrhizal fungi. So then he ground them up, and he did the amplicon, PCR amplification of ITS and sequencing. So ideally, this is what he would have gotten back, right? He would have gotten back from his samples the same number of, or proportion of reads that would have matched the number of reads that he put in. But I'll show you the first one, the best one first. So you can see that they didn't come back in the proportion that he added them. Okay, so there's some effect of too much template or too little template that influences the number of reads that you get back. And then if you looked at the other fungi, they didn't come back in proportion at all. Some taxa, either the spores didn't break as well, the DNA didn't isolate as well, or it didn't amplify as well. So this is another problem that ecologists have. They'd love to know actual abundance, but they can't get that information yet. So maybe that's something systematists could help with. And then he goes back to this problem about the equating ecology with the operational taxonomic unit. Even with better algorithms and databases for taxonomic assignment, we still know very little about the detailed ecology of even OTUs for which a clear taxonomic assignment can be made. And then difficulty in assigning trophic guild, in other words, what they're doing in nature, how they're eating, is not an uncommon problem, despite the fact that trophic mode is perhaps the most fundamental feature of an organism. And so I want to go back to my Neurospora example. So Neurosporas are pretty much fire-adapted tree associates, and Sordaria are dung fungi. So they have very little, very different trophic features, but you'd get the same, you'd think they were the same thing with the ITS. So again, here's something where we ought to be able to help ecologists. And then the other problem, and this is, we work on indoor air, some of the projects that I'm working on with Tom Bruns, and we collect spores, and then we sequence the DNA, but we don't even know if they're alive, let alone what they're doing. So that's another problem that ecologists have. So, and an example of that is New Nguyen working with Tom Bruns and Nicole Hinson. Tom has a project where he's buried mycorrhizal, spores of mycorrhizal fungi in several aliquots in soil at Point Reyes, with the idea that he's going to sample them, or someone's going to sample them, over the next 100 years to see the longevity of these spores. So we know they're just spores, and they assay them by putting the roots of pine trees in to see if it forms mycorrhizae. But New also just sequenced some of them straight from soil. Sure enough, had no problem retrieving all those spores, and we know that they're not active. So that's another problem. So after that depressing start, I want to talk about some encouraging history of the use of the sort of the data that systematists have provided to ecologists. And the first one that I know about is this another paper from Noah Fear's group. And this was one of the first metagenomic studies. They called it metagenomic, but it's PCR amplification and sequencing. And they did it with Sanger sequencing of cloned PCR products in 2007. And basically what they discovered was there's a tremendous diversity. And so this was a figure from their paper. And that line there, this probably has a pointer, yeah. On that line, for every time you added one of their Sanger clones, you would add one new fungus. Okay, so in other words, if your sampling followed that line, every time you looked at a new clone, it was a new fungus. You never found the same fungus twice. That tells you you've not sampled enough. Okay, so you can see that in this early study, if that's the line, they didn't sample enough. And down here, the organism that was sampled the most was desert fungi, I think. So the fungi were actually sampled a little better than the archaea and bacteria, but clearly this was not enough. Sanger sequencing was never going to do it. So then we'll go to one of Ari's papers in 2012, where they're now using next generation sequencing. And here's that same type of plot to see if you've sampled enough. This is the line that was from Noah's paper, where every new sequence would give you a new OTU. And you can see that with next generation sequencing has been a huge improvement in getting enough samples to actually begin to think that you've sampled the diversity of the organisms. Okay, so that's good news. We're not there yet, but we're getting there. But how about now activity? Because activity is what ecologists really are interested in. So there's been some advance in this. So this is a paper from 2012 from a French group, and they worked on two forests, beech forest and a spruce forest. Again, it's Sanger sequencing, really heroic work. So they looked at only, however, 96 18S RNAs to try to identify the organisms, and they used ribosomal RNAs, so they were getting presumably living fungi instead of perhaps dead DNA. And they looked at 10,000 mRNAs to look at function. And I'll show you one figure that I think is interesting from the paper. So this is the cDNAs made from ribosomal RNA. So these organisms are alive, and they looked at everything in the soil. every eukaryote in the soil here. So there are plants at the top, and then animals and fungi. And then if you look at the mRNAs, you see that the fungi now represent a bigger chunk. And mRNA would tell you, transcripts would tell you that they're active. So we know now that the fungi are the most active group in that environment. Now what's interesting is we think that the enzymes that fungi make that break down plant cell walls are sort of the province of the mycological world. But this study showed, for example, here is one of these endoglucanases, the G45 endoglucanase family. And sure enough, there are a lot of fungal genes that they found, but it turns out, and who knew, mollusks are making a lot of these too. So fungi aren't the only organisms in the soil that are breaking up the plant cell walls. Okay, another more recent project, this one is from 2014, published in 2014, somewhere there. And this is one that Tom Bruns and I and Ritas Vilgalis and Kabir Pei have been involved in. And here, the focus was to try to narrow the diversity. The focus was on pine forests across North America. We threw in one spruce forest because there are no pines in Alaska, and we wanted to sample in Alaska. And there were 25 forests were sampled, and 600 soil samples. So in the ecological studies, sampling is a big deal. You have to have a lot of samples. Illumina sequencing this time of ITS, which I see now in the literature called ITAGs. And then Kabir analyzed the activity of 7 enzymes. So to look at activity, he looked at enzyme activity rather than transcripts. And what's interesting about it, the main message, is that, so here's a map. The color of the parts of North America correlate with the color of the spots on this graph. And so the fungal taxa here sort into geographic communities. So for example, there's an Alaskan community. That was an interesting thing. We weren't sure that that would happen. And that sorting into communities supports a claim that community similarity drops as spatial distance, as geographic distance increases on this log scale. So there's a distance decay, which we commonly see in microbes. What's interesting, though, is that Kabir's enzymes did not sort geographically. Instead, they were more correlated with the environment of that particular soil, especially water availability. So the answer seems to be that fungal communities in these pine forests are assembled from different taxa in different parts of the country, but they do the same thing. So they have the same function. So that was an interesting result. More recently, Sunny Liao in Ritas' lab, again part of this consortium with Tom, Ritas, and Kabir, looked at metatranscriptomes. So she added, looking at RNA, to what Kabir had done with the proteins. And here we've restricted it even further, not just to pine forests, but to one species of pine, because it gets more complex as you add transcriptome. And there are 2 sites in the pine forest in the southeast. At each site, just 2 soil cores, and from those cores, 3 root clusters and 3 soil samples are what Sunny looked at. And so first, I should go back one. And so she looked at DNA, ribosomal RNA, and mRNA. So this would give you all the taxa, including the dead ones. This ought to just give you the living ones, and this ought to give you the active ones. So this is the DNA, and this is a mycorrhizal study, so I'm sorry there aren't any ascomycota. Well, actually, that's not true. There's amphonema, so, and tuber. So this is a variation with the DNA. If you go to the RNA, there's less variation. There are fewer taxa. And so there are fewer taxa that are alive there than ones that just have DNA, as our inference from that. And then you'll notice piloderma was common, and piloderma had been sequenced at JGI, so we went with piloderma because you have to have a genome sequence if you're going to do metatranscriptomics. And so here is Sunny's figure, and she's got the root samples and the soil samples, and then as you go around this circle, you go from, for example, enzymes or transcripts involved in translation. That would be the first, and there's a big unknown group. then various functions. And if you look around here, the colors are found in all of those samples until you get clear to the bottom here. So I'm just going to emphasize those two today. So most transcripts are shared by the fungi in the soil and in the root. Except if you get to the fungi in the roots, they have an excess, here I guess that's a purpley color, purpley pink, of secreted proteins. And that we think that these may be effectors, like Joey was talking about, proteins that fungi make that help them manipulate the host inside the mycorrhizal root. And if you go into the soil, instead of the roots there, you see an excess of carbon, nitrogen, and phosphate transport enzymes. And that could be involved in the business end of the mycorrhiza, where it's acquiring maybe not so much carbon, but nitrogen and phosphate from the environment. So this kind of study, this is a very early primitive one, shows that you can look at function of fungi in different parts of the environment if you pick a simple environment. So the consideration of activity requires sequenced genomes. And so Joey, I don't fault Joey for his plug at all about the 1,000 Genome Project. And it's a great thing, it's just not enough. We need more than that. So now I'm going to go to the third recent review on this topic, one that David and Jason Steitch and Joey wrote. And to emphasize the need for the genomes, because the ecology, if it's going to really move into function, we've got to have lots of genomes, genome for every fungus that's in that environment. And so they wrote, it's not unreasonable to think that in the near future, mycologists will routinely sequence the complete genomes of isolates that are the subjects of their research, much as ITS sequences are now used to confirm identifications. And that I want all you to hear that, especially Pedro Kraus, who's here with this huge collection. That would be lovely for that. And then, I forgot, I guess there are 4 recent ones. And then Cheryl Kuski also is the lead author on another recent sort of position paper. And in there, they've got a figure. And the figure shows, talks about community complexity and sequencing diversity and sequencing survey size and thinking about genomes at the bottom. So if you're interested in one fungus, you only need one genome. If you've got a constrained system, like a host pathogen system, the mycorrhizal system with one tree that I talked about, maybe animal rumen or really extreme environments like mine drainages, you may be able to get by with maybe tens of genomes. But if you want to look at the communities that probably most of us really care about, then you're going to need hundreds to thousands. And so the black line's what you need, and the blue line's what we've got. So we need more genomes. So I talked a little bit about range maps, Noah Fear's range map, and then I've talked a little bit about function of fungi in natural communities. And now I want to talk about the last thing, function from experimental studies of community function. So the next thing in ecology, past seeing what's happening in nature, is to try to do experiments and see if you can test some ecological theory with these experiments. So function from experimental studies of community function is going to need a simple community of fungi that you can cultivate, because you've got to be able to play with them in the lab. And so the first studies of this are on cheese. Who knew? So first of all, there were several groups that were studying what sort of fungi are in cheese, because it really wasn't known. And of course, one of them, of course, would be in France, but there are several others. But what I want to talk today about is the first of these experimental studies, and this is from a group at Harvard. Ben Wolf was the first author, and it's in Rachel Dutton's lab, and it was published just last year. And so they looked at 3 kinds of cheese. They call it bloomy rind, natural rind, and washed rind cheeses. So that would be like Camembert, Saint-Nectaire, and Époisse, something like that. Now, the cheeses that they work with aren't they're not going to fill you up at a wine and cheese. So they make the cheeses in 1 milliliter wells and 96 well plates, okay? But that allows them to replicate, which it has massive replication, and they can play with different components. So for example, here, these are the fungi across the top mixed with just the fungus alone, or plus an arthrobacter. So they can do experimental studies to see the effect of different microbes on the end product, the cheese. So first, of course, they did an ITS study of cheeses from all over the world. And they found that, so this, and I can't figure out from the paper, I'm going to have to ask Rachel, but one of these is bloomy rind, one of these is natural, and one of these is washed rind. And there are bacteria on the top and fungi on the bottom. But the story here was that geography doesn't matter. If you make the same kind of cheese in different parts of the world, you have the same microbes in the cheese. Part of that's because you buy starters from the same companies, but part of it, those organisms didn't come in the starters. They're just in that environment. You've created that environment. And so here are the bacteria and here are the fungi, the most common ones that they've cultivated. So they've got everything that's more than 1% of the fungi, of the organisms. And you can see the bacteria are quite variable, more variable than the fungi. Fungi in some of these cheeses are remarkably stable all over the world. But what's really cool is they can look at succession. So they can start the cheese and then sample and see what the microbes do. So each of these batches was started with a mixture of all of these taxa. So here's just bacteria alone, or the fungi, which is the one I want to concentrate on, 3 fungi. They were all put in at the start, but at the start Candida took over. And it took a few days for Candida to work on the environment until Penicillium could come in, and then Scopulariopsis. And this is just 3 replicates, so you can see that it's replicable. So it's a very interesting system to study basic ecology. Of course, it requires genomes. They have to have a genome for every one of these. So the last thing I want to talk about is political considerations. So fungi are essential environmental players. We know that, okay? There will never be enough mycologists to study the fungi in the environment. There just will not be. So we have to make it easy for ecologists to include fungi in their studies. And so, you know, it would be if a zoologist was going to study some animal, the ecologist some animal, and ignored the plants, they wouldn't be able to get the study published. We have to make it so they can't ignore the fungi. They wouldn't be able to get the study published. And so how to make it easy to include fungi, or why? So I want to show 2 publications from last year that highlight the problem. So here's land use type significantly affects microbial gene transcription. And so they did a metatranscriptic study, forest versus grassland, and they were interested in organisms and enzymes that degrade aromatic ring compounds and that degrade lignin. So they surveyed the microbes there, and you can see there's bacteria, archaea, eukaryotes, and in the eukaryotes, they're the fungi, plenty of fungi. Whoops. They didn't study them. They didn't include the fungi in a study of aromatic ring compounds and lignin degradation. Okay, probably missing something in that study. And you know, some editor should have said, wait, you have to study the fungi. Here's another one, metatranscriptomic analysis of Arctic peat soil microbiota. And so the enzymes here are the enzymes that we all know, the carbohydrate enzymes, the CASI enzymes, oxidases, hemicellulases, cellulases, debranching enzymes. These are the enzymes that are made by Trichoderma and other Ascomycota commercially. And here are the microbes that they used in this study. There are some archaea and there are bacteria, no fungi in the breakdown of peat soil. So I wasn't sure about peat soil, so I emailed Lynn Boddy. I said, would fungi be expected to break down peat soil? She said, absolutely. So again, we're missing out. Fungi are not there, so we have to do something about that. And what we need to do that is to make it easy to include fungi in these studies, and what we need for that is lots of genomes. So I talked about how systematics serves all other sciences. We believe that, especially today I talked about ecology. Massive sampling and range maps, I think, is going to raise the visibility of fungi. I imagine those maps are going to be very useful to allergists. move from composition of communities to the function of communities. And for metatranscriptomics, which tells you if things are alive and if they're active and what they're doing, which is what people care about, we need genomes. And so the aim is to enable all ecologists to study fungi. And I thank you very much.
Thank you, John. The floor is now open for some questions. Please use the microphone.
I'm very interested in your sampling techniques, actually, John. Could you said in some of the things, I believe, that 1,000 samples have been taken. What I'm interested in is, how do you take your samples? Do you differentiate at the depth? And what's the spatial relationships between your samples?
Very good question. So at each site, we selected woods that had only one species of pine. But at most of the sites, we were in a region where there were several species of pine, at least two. So we would pick stands of species A and species B. In those stands, we lay out a grid that's nested squares. So for example, it would be 5-meter square with a 5-meter side, 10-meter side, 20-meter side, 40-meter side. We sample on those sides, and then we sample the diagonal. So we have the potential to make many different distance comparisons within that sample, and then we can do it between that grid and the next grid, and then between that site and the next site. So there's a hierarchy. Then in terms of depth, we started out sampling the organic horizon, the litter, the organic horizon, and the mineral horizon. Turned out the organic and the mineral, there was no difference in these soils. They're montane soils. So then we ended up just looking at the litter and the soil.
At what sort of depths, though? And with your grids, what sort of distances? Are you talking centimeters, tens of centimeters?
No, the nearest sample would be 5 meters apart within a forest, and the depth in these forests, we didn't go down much further than that because it's really hard to go down further than that in montane pine forests.
Yeah.
And you get through the organic layer very rapidly.
Yeah, I would say that's incredibly crude sampling, so you've proved your point, even by describing your sampling. You need vastly more samples, because in my experience, it'll vary enormously, even in your grid.
Well, it depends on how homogeneous the environment is.
It's not homogeneous. It won't be homogeneous.
No, I agree with you, but okay, so maybe two comments. The first is, that's absolutely right, but the common things will be there. As the tail goes out and you get the rarer things, they're completely different in every sample.
Yeah, okay, thanks.
More questions?
I was just thinking about what it means to be dead. So if you have a mycelium, what is the process of dying? And is it the case that once it's dead, it doesn't come back to life as quickly as it would if it were growing from a new stock?
That's a really good question, and that's one that I really can't answer. And it figures in things like lawsuits. The wood got wet when one insurance company had the policy, and then they switched insurance policies, and the house was rotten, and they want to go back to the first carrier and go, well, it started growing from the minute that it got wet, and there are big legal fights over that, and then they say, no, it dried out in between, so the fungi died. I don't think people know. And like, for example, wood rot fungi, what happens when it dries out? Do they die, or do they sit there and then it gets wet and they start again? Anybody know? Yeah, that's something that can be discovered now.